Mike, okay, yeah. We have our star guest here, Joseph Powell, to drive the single-cell genetics breakout session. Hope to have participation from all of you. Our main goals here, as always, for any, I don't know if we'll let people. Is it possible to close? Okay, we'll wait a little bit and then we'll close that door. Our main goals for this are the same as for any Human Cell Atlas Asia breakout session, which is to nucleate. Where's Bridget? Thank you. Is the Zoom on? Yeah. Okay. Our main goal is to nucleate transnational collaborative work within HCA Asia, also to nucleate new teams contributing data to the global human cell atlas, and then building those relationships and connections. So hopefully you will all speak up during this breakout session and discover each other and identify your your partner for your next big study, right? And with that, I'll hand over to Joseph to drive.
All right. Whoa, that's loud. Hi, everyone. We've got the kind of slightly weird space here because it's the combination of we're sort of sit here and it's also the thorough affair for other people. So if you're partaking in this session, can I encourage you to come take a seat so we sort of know who's who in the zoo? And if you're not, just listening with a bit of jealousy. So for those of you that I've not met before, my name's Joseph Powell. I'm a geneticist at Garvan, do a lot of single-cell QTL genetics. We had a few things we were keen to sort of just guide people through and have a discussion around areas that we thought the community could come together to support collaborative projects and activities for HCA Asia and HCA more generally. But I'm just going to do a few introduction slides for those of you that are not super familiar with the intersection of population genetics and cellular genomics. Before I go through this and just embarrass myself with naivety, but can I just get a show of hands? Who has worked on some form of actually not start single cell, EQTL or MQTL bulk genetics? So I have, Drew has, Ang Lee has. Put your hands up so we know. All right, so good representation. And specifically single cell genetics. Our same people. Oh, great. So we've got captive audience. Fantastic. All right. So we don't need to labor this too much, but for those of you that are not super familiar, the general concept that we're interested in is there's genetic variation that exists between individuals. There's lots of types of genetic variation, common SNPs, rare single nucleotide polymorphism, structural variation, and using single cell and spatial technologies, we can resolve how that variation contributes to molecular and associated phenotypes at the level of cells or cell states. And there's a lot of really cool stuff that you can do beyond just looking at how does it change the mean expression. You can look at how does it affect the variability within a donor or between individuals. You can look at how those genetic effects vary as cells transition across different state spectrums, which is an interesting area of methods development. But in general, the way that, I think most of us have been working on this, I guess, is that there's a few things in common. We need cohorts of data to capture genetic diversity and genetic variation more specifically. And that is particularly relevant for statistical power. So to do that, obviously, we need to generate a lot of single cell data. So we need scalable approaches to do that. And that's typically come through, I guess, using genetic multiplexing approaches. And again, just in the sort of show of hands, can I just see who's generated single cell genetic data and used like a multiplexing approach? Hands up, multiplexing. Hands up anyone that's generated data where you've done one biosample per kind of reaction. All right, rich. And then obviously there's some interesting components of methods development as well, whether it's the mapping models or identifying cell states and so on and so forth. And I guess, so I'm a geneticist by background, and, for us, we're particularly interested in this because we want to link it to higher order human variation, disease genetics, or other forms of variation. And so, the standard way to do that is to use causal inference approaches to overlay known genetic risk factors, GWAS, for example, with single-cell EQTLs. Sorry, my wife's trying to call me. Oh. She does know a little bit about genetics, so she could join. Yeah, and so obviously this has been a growing area in the field. This is a figure that came from a review article or perspectives article that we published last year. So it's a little bit out of date, but this gives you an example of, I guess, where the field started. Sort of 2018, the first papers were published linking genetics and single cell gene expression. And then there's been obviously, increasing number of publications more recently. And then with that, an increase in sample size, largely, an increase in both the number of donors, but also typically the numbers of cells per donor. And then there's a series of papers that have come out, again, probably some from you in this room since, sort of late 2022, more recently. I think, and this is particularly relevant for what we might want to get out of, this community efforts, but there has been a global effort. So I'm guessing some of you might be involved in this, a single cell EQTL consortium. Is anyone actively involved in this? We are as well. A few people. Great. So this is basically a consortium to bring together studies and create a couple of things that are valuable. I think one has been a series of sort of standardized analysis approaches and software that can be used across different groups. That I will say I feel like that you need to continually develop these methods, and we shouldn't assume this is set in stone and we've solved the problems of the analysis frameworks. But the single cell EQTL consortium has produced, I think, some quite nice pipelines that are accessible to the community for looking at QC, demultiplexing. Drew led that effort. Single cell annotation, and then some approaches for looking at genetic effects. And it's also brought together some cohorts in the community for meta-analysis as well. So I think there's about 14. Shyam, do you know exactly 14 or so cohorts, sub cohorts as part of single cell EQTL gen? More?
Don't remember.
All right, it's 14. Let's make just it's fact now. But this is almost entirely in PBMCs, and so there's definitely an important question in my mind, and hopefully I'm keen to sort of draw people into their views on this, of whether or not this can be extended, or these approaches to different tissue systems and how we might go about doing that. And then of course there's some interesting components of methods development and making comparisons across different cohorts and understanding the contributions of genetic ancestral diversity and environmental variation and how these things contribute to variation in the cohorts that we're generating. So these were the sort of introduction slides I just wanted to quickly walk through, appreciating that some of you are very familiar with it. And then what we were keen to do is just have hopefully a pretty dynamic conversation around a few topics that Shyam and myself had thought might be interesting for the community to engage in. We're of course very interested if anyone else has other views that they would like to discuss. And because it's kind of a noisy room, if people are quiet, I might just start calling on people to contribute. Those of you that put your hands up earlier, so it was a trick. So before I switch into some of the questions, does anyone have any questions about sort of the background and what we want to get out of this interactive session?
Two quick questions about the single cell EQTL mapping. Usually, how large for the sample size is required? And the second question is it specific for normal tissues?
Yeah, good question. So, I mean, sample size is an interesting one. I mean, my view as a statistician is basically more the better. But you sort of, I mean, and that basically, to what degree of effect can you identify, basically? But I think once even the allelic effects for these things tend to be quite large, right? And so even once you have tens of donors, like 100 or so, you can start identifying plenty of single cell EQTLs. Obviously, you're going to miss a lot of true effects just for underrepresentation of power. But the other thing that I don't think we've really fully addressed as a community yet is what's the effect of cell like numbers of cells of a certain cell type or cell state, that definitely impacts power. So if you look at a lot of the blood studies that we've done, you have, same donor sample size, but the power to detect effects drops off with the frequency of a cell. So it's like, if you think of data generation, it's a completely like for us, almost competing priority is you've got, and X dollars to spend, you're going to spend it on more donors or more cells per donor. And it's kind of a trade-off, in my view. I don't know if anyone else has got a different perspective on that.
It's a great question, Dan. When you designed the study, to be honest, when we designed our QTL analysis, we were just guessing. We said Joseph had done 1,000 cells per donor. This a few years later,
we
we'll
guessed
do
as well.
2,000 cells per donor is 2x.
Yeah.
And of course, you want to use the latest reagents. So then from the same number of cells, you get more QTLs because the data are cleaner. And then how many donors was just like how much grant money we had. So pure guesswork. But with 2,000 cells per donor and 600 donors, we ended up getting E genes, QTLs for, I don't remember, like close to 10,000 genes. So let's say roughly 10,000 genes if you add up across all cell types. So that's a pretty healthy haul of QTLs just from 2,000 cells per donor. And with the slightly, the previous generation version of 10x genomics reagents. So now with the GEMX, which is more sensitive, so you get cleaner data per cell, I think you'll get even more QTLs for a cohort of 600 people. So I'd say 600 is quite healthy, but with Joseph, your data, I think it'd be a very interesting scientific question to say number of cells versus number of reads per cell versus number of detected genes per cell versus cohort size. Wow, what are the big factors? How can you estimate how many E genes you hope to get, or your statistical power, something like that? I don't think anybody has done that in the single cell field. At least for us, we're all just guessing, rule of thumb, that person succeeded, okay, we'll succeed too if we do slightly better, that kind of thing.
Yeah, I mean, that's actually quite an interesting thing because, I was saying you could toggle the number of donors, numbers of cells per donor, but you can also toggle, how deep you sequence, whether you're doing short read, long read, there's a whole bunch of factors that are all going to contribute, right? Yeah. Has anyone worked on power calculations for single cell genetics? Yeah. Can you share your views?
The number of cells hi, I'm Ryu from Japan, and I'm calculated how EQT power was influenced by the number of cells per sample and the total number in the cohort, and both important. But sample number, sample number is more critical. And when sample number is less than 100, EQT power is dramatically dropped. If more than 100, maybe like this, and less than 100 too. But also the number of cells per sample is very critical to EQT power, to detect the EQT power. Yeah. So we demonstrated in my cohort and 1K. Yeah, and it's very similar.
Yeah.
My paper is power analysis. How
have you published it yet?
No, not yet.
All right.
Yeah.
Keep our eyes out.
And reviewer required us to evaluate our data set and 1K.
Yeah.
Yes.
Okay, that's exciting, Rhea. I think it'll be a real service for the field, just for the group. Rhea is one of the leads in Yuki Okada's group, formerly at Osaka University and now moved to Tokyo and Riken. Yeah, so ping him if you want to design your single cell genetic study.
Okay, just to add some historical context to the question as well. Introduce myself. Oh, my name is Bo Xiang. Hello, everyone. I'm an assistant professor at National University of Singapore. So before like the single cell field really bloomed up, people have done QTL for 2 decades. And so I was part of the GTAX consortium, and we worked extensively for over, a period of 1 decade on this pen tissue QTL project. So in 2012, there was a GTAX paper, that white paper that analyzed how many samples you would need to achieve certain power, right? And then the rule of thumb for bulk tissue is about 60. And there's a few reasons for that, right? So we made some assumptions about the effect size and also the variance. So let's say that you have a couple of samples, you can estimate, like the average effect size for some well-represented genes. And then you can also measure the variability of some well-represented genes. And using these two parameters, you can do simulations. And then let's say that you aim for 80% power, then usually you get, somewhere around 60. So that's argument number one. Argument number two is your minor allele frequency, right? Say that you have 60 people, then you have 120 alleles. And usually we use a minor allele frequency cutoff of 0.05, right? So 120 divided by times 0.05 is about 6, right? So in order to have enough replicate to have to be confident in your estimates, you probably need more than 3. So 3 to 6 is a healthy range, for your minimum sample size for a specific genotype. That's why people settled on 60. But then after we moved to single cell, I think Rio is absolutely right. The number of samples requirement has increased for a couple of reasons. The first one is that not every single cell type would give you enough cells for you to do a pseudobulk, right? So let's say, some rare cell types, maybe you have 100 people, but then only 60 of them has sufficient cells for you to do a pseudobulk, right? So you have to account for that. And also in our hands, we realized that the effect size that we can capture in the single cell level is generally smaller than bulk for good reasons, right? Because bulk, you have higher sequencing depth. So for those reasons, yes, you're absolutely right. I think we need at least 100 per cohort, and then accounting for all those, variability, lower sequencing depth, dropouts, and also rare cell types, and you get somewhere around the same power as you doing a bulk study, right? So, but also I concur with Joseph and also Shyam. The money is the limiting factor. We want as many samples as possible. So that's just some historical context.
Yeah, that's super useful. Thank you. And there was another comment about what type of, let's say disease states you were asking about, or again, I'm kind of keen to kind of survey the community here. I mean, we've largely, I mean, the latest cohorts that we're generating have a combination of control, and when I say control, I mean they're like normal individuals, right? So they actually have all sorts of different conditions like a normal population does. So if you can capture clinical metadata, you can do some really interesting stuff with control populations. But we've been generating cohorts where we have controls, and then we've got cardiovascular, autoimmune, cancer, so disease subtypes, largely of PBMCs, a couple of solid tissues, but not too much. But out of interest, those of you that have been working in a space, who's working on control/healthy cohorts? And yeah, most people. And anyone working on disease-specific cohorts or case control? So could people shout out what are those disease groups they're working on? I'm interested. We might start on that side and sort of. Oh no, we're going to start here because Shyam's given the mic.
Liver, definitely.
Liver, all right. Ah, cool. Yeah. Yeah.
Type 1 diabetes, glioma, liver. COVID-19 and
Yeah.
autoimmune disease.
Yeah. And
Sorry, so these are very valuable because I'm hoping that other people are working on similar things. So I'll just repeat again, COVID-19, autoimmune disease, including type 1 diabetes, liver, glioma, and we are doing type 2 diabetes, and we have done COVID in the past. Any other diseases people are doing QTL analysis on at the moment, in future? Okay, so it
That's
looks like
useful. Thank
diabetes is a major theme and autoimmune disease is a
Mhm.
major theme, which makes a lot of sense. Even people who are not in this room are doing autoimmune disease, single cell genetics of PBMCs. There's quite a few people, so
Yeah.
that might be a point of convergence.
Yeah, that's super interesting. Thank you. So as Shyam mentioned at the beginning, one of the key things of these breakout sessions is we're keen to have a discussion about what, opportunities there are for the community to come together and to collaborate. We were walking from the hotel and put together a few, I guess, key questions that were in our minds. And, we were keen to have a conversation about this, but also flag if there's other things that people have been thinking about that are particularly useful for the community, and opportunities from stuff that we're either currently doing or, we can, planning to do in the future, then, then it'll be really valuable to have a discussion around this. And I guess just to sort of start, like the kind of the three things that were most interesting to us, were, a lot of us are working in easily accessed tissues, circulating immune cells, stem cell systems, so on and so forth. But there's and I think, was it Drew or someone else showed a slide yesterday that had a sort of a breakdown of, I think it was in one of your slides, Drew, actually, maybe someone else showed the same thing. It was a breakdown of the different tissues that are represented in the human cell atlas. And there's a genetic component. So there's lots of tissues represented, but in the genetic component, it was still predominantly immune cells, perhaps for no surprise, because we need to get large sample sizes and they're difficult to collect. But we feel this is definitely an interesting opportunity. And so moving into tissue systems that are less easy to collect, and also the sample prep is challenging. Can we think about opportunities to look at spatial genetics, where if we're generating spatial data, where you can, on at least cohorts of data, can we collaborate around the role of genetics in cell types or cell interactions using spatial data? So that's one, and we can maybe just whiz through these and have discussions about people's interests and what the opportunities might be. But the other thing that's quite apparent, I think, with things like ADA, some of the work we're doing, and I think some of the stuff that other people might be working on, that, we have actually a pretty rich representation within the Asia Pac community of cohorts that are developing, again, predominantly on PBMCs. And so there's some really interesting outcomes that can come from looking at integrating data across those consortium, and how we can evaluate, what's the factors that contribute to differences in expression in cells, whether it's genetic ancestral diversity on its own causing variation, or an interaction with environmental factors, let's just call it geographic region, and separating that from just technical variation of us generating data in different labs. So that might be an interesting opportunity. And then, from a practical perspective, we thought that it was important to think about sample access for future between country consortium efforts, and what the barriers and boundaries there would be to establishing that. And then these were just other interesting questions, as you can read, that we thought were kind of, like that there might be energy within the community to come together to address. I won't read them out because you can all read. But on those first three, maybe I can just ask, does anyone have any other views of things that the community could come together to help solve that's valuable for the broader efforts of the Human Cell Atlas that's not captured here? Or are we just clairvoyant and we've figured out all the best questions?
Okay. So apparently you're one of the leads for the EQTL consortium, and you flashed a slide before. I think one of the most challenging problem in these across cohort effort is the data normalization and also federated learning. Can you just perhaps say a little bit of how you do the data normalization, and also, because I imagine different subsets of cells might be captured across different cohorts. And also, like, how what do you mean exactly by doing federated learning?
Yeah, so this was a particular challenge within that community, in that in many instances, the raw data couldn't be shared between cohorts, certainly at the time. I mean, some cohorts can share the raw data, but most of them couldn't for a combination of both practical and ethical or ethico-legal reasons. And so the approach that the single-cell EQTL consortium has come up with is that the meta-analysis is largely operating on the summary statistics. So to get to that point, so in other words, every cohort runs the same pipeline, gets summary statistics, and there's a meta-analysis across the various sub-cohorts where you weight the summary statistics based on the sample size and a couple of other factors of a sub-cohort. But of course, to get to that, if you were just to take all the summary statistics that have been published, they all would have had different cell classifications. So you've had the issues we talked about. There have been different normalization, different QC approaches. And so the federated, harmonization, as it's referred to, is basically how do we standardize those pipelines so that, we rerun them on each of the sub cohorts so that the summary statistics are as generated in as representative as way as possible. You will still get things like differences in cell proportions between sub-cohorts, but you can address that using weighting and the summary statistics largely. But the key things were, classification of cells using individual annotation of cell subtypes. So you're not relying on clustering, which is going to have biases between cohorts. Using the same approaches for demultiplexing. So again, you're not missing kind of classes of variation that might happen in different cohorts. And again, the same standardization of normalization so that if you're correcting for batch effects, you're kind of hopefully we're all inducing the same biases or hopefully removing the biases as the case may be. Drew and Ang Lee, you guys have been involved in some of the work there. Is there anything I'm kind of missing that, yeah.
My name is Ang Lee. I'm a postdoc in Garvin Steel with Joseph Powell. I think one of the aspects I can think of is the disease-specific EQTL. So if you look at all the GWAS paper in the last 2 decades, when people have the 1 million, 2 million GWAS and then start to integrate with the EQTL, they all use EQ Gene Consortium or other GTECs, most are from the healthy individuals. But there's a lack of disease-specific EQTL. I think one of the reasons that it's really hard to collect those tissue and have a large scale EQTL. And I think one of the things is missing that if we have a equal power healthy EQTL cohort and disease specific cohort, how much carbon power can we gain to integrate those signals? Let's say if we have 1,000 individuals from healthy individuals, and then you try to integrate the 1 million type 2 diabetes GWAS, and then if you have a 1,000 individual with type 2 diabetes, you get EQTL from blood and also from ideally pancreas, but maybe not within the 3 years or 5 years. But then you compare the signals they can identify. How much can we lose? Will we lose maybe 30% or 40% or even more? I think that's something that we're missing here. But we do need money to collect more samples.
That should be just an asterisk at the end of every comment, shouldn't it, Ang Lee?
We need money.
Yeah.
Hi, I'm Ryu from Japan, and I have a single-cell EQT analysis in COVID-19 and healthy, and we also check the induction QTL versus COVID-19 healthy, but I found that it's not work well. So because in COVID-19 and healthy, maybe in average, maybe have a different gene expression, but not all of the cell, just average. So what I think that so dynamic QTL as a context like a cell state, so dynamic QTL analysis using a cell state, and I think that what is more power to detect such a kind of a localization. Yes, and we found that such a dynamic QTL analysis are more likely to be enriched in enhancer lesions. So yeah, I think that is one of the important thing. Yeah, not just this interaction.
Yeah,
aggregate and dynamic QTL.
Yeah,
in many cell states concerning. Yeah, yes.
Yeah, because I guess probably a few of you are finding. I mean, there's plenty of this published, but in many instances you don't have a disease cell type and a non-disease cell type, but you have a shift in some genomic profile that's quite subtle, often within a particular gene program, right? That's either a cause or a consequence of disease, and that helps. If you can put those cells in that cell state that's defined by that biological phenotype, then you can do those dynamic effects. So
If I can pick
Sorry,
up on that.
go on.
So that has come up a lot even before single cell genetics, that you want to perturb your cells and bring it close to the disease state in order to learn something about the genetics of that disease. So I guess you're assuming you're saying, Ria, that you want to do the disease specific or disease state dynamic QTLs. Have you found empirically that for example, COVID GWAS, have you found empirically that the COVID dynamic QTLs are more useful than the healthy dynamic QTLs for co-localizing COVID GWAS? Is that anecdotal or statistical conclusion? Because there's not that many COVID GWAS loci anyway, right? So it'll have to be an anecdotal conclusion, I presume, right? Okay, but still a very plausible one. Yeah.
for those on Zoom, we're getting nodding of yes to Shyam's question.
If there's any questions on the Zoom, I encourage the people on Zoom to please type out their questions, and Bridget will read them out for the audience. Anyone else have this question here?
Okay, hi, I'm Yu Shi from the University of Science and Technology of China. And I'd like to know, we all know through QTL analysis, we could identify numerous e-genes. But how can we further explore the role of those e-genes or the regulatory relationships in the context of disease mechanisms?
Yeah, I share my perspectives on this. So I think there's a couple of ways you can do it. I mean, if you have cohorts where, like we've just been describing, where you've got, like, let's call it phenotypic information on the individuals, you can start, I think, separating them by cell state and, like we've just described, and start looking at how those variation occurs. But from a statistic, like from the human genetics statistical perspective, we already know a lot about genetic risk for most complex diseases, and actually plenty of rare ones as well. And most of that actually changes in genome regulation. So you can use causal inference approaches, for want of a word, like co-localization, Mendelian randomization, to link known segregating genetic risk for disease through to changes in genome regulation of a particular gene in a certain cell type or cell types. There's definitely more that I think can be done methodologically to improve that. I will say, I think, like, at the moment, most people, again, I'm guessing, are doing single cell RNA sequencing and single cell eQTLs rather than chromatin accessibility, and it's emerging, or other forms of epigenetic and variation and particular mechanisms. So I think we're still at the foothills of figuring out how can we, we've got DNA changes in expression, but how's that functioning, acting? Is it acting through changes in transcription factor binding in a cell type specific manner? Alternative chromatin accessibility, so on and so forth. So there's going to be a lot of work, I think, that'll have to happen around building the right maps and data for non-single cell RNA sequencing to properly get to some of the actual causal mechanisms, for want of a better word.
Okay, thank you.
Don't know if anyone else has a perspective on that.
Thank you.
Excuse me, I'm her colleagues. I'm a postgraduate from USTC. My English is not that good, so I didn't catch the questions you just discussed. You just discussed. I don't know when there is a disease cohort and a healthy control cohort, if we should analysis the single cell eQTL respectively in each cohort, because I think theoretically there is different regulating patterns in different contexts. Yes, this is first question.
Yeah. So I mean, like, other people might have again something they want to chip in around this. My views, I guess, it partly depends. The exact analysis approach somewhat is dependent on what exact question you're interested in. Most diseases that I certainly am involved in working in, like common complex diseases, for want of a better word, I think it's better to analyze the cohort, as a whole. I mean, the worst, let's say, for example, you've generated all your cases in one lab and your controls in another lab, then, that's a challenge, right? But if you've generated them collectively, then, having analysis as a single cohort where you fit some form of covariate factor associated with clinical information, or even better, you use that to help define cell states and variation. I think you'd be better powered to possibly unpick some of the mechanisms of how that disease is ultimately contributing to variation in cells. Again, I don't know if people are working on this that have something they want to chip in. The people who are working on disease cohorts, who has sort of case control designs? Those people.
Yes, I think GWAS is done between disease and healthy controls. So I think the disease associated GWAS loci, I think it's better to be done in a disease cohort.
It's definitely more advantageous. Yeah, I mean, you'll have better power. Yeah.
Yes, and another question is, I've read some single cell EQTL passages. Their work mainly focus on mathematics or statistics and calculate how many EQTL loci are common or specific in different contexts or different cell types. Some immunologists may not believe this kind of results, so they want some validations such as experiments or something else. How do you think about this?
I think I get that question from my immunology colleagues a lot. I mean, I'm a geneticist by background. I don't want to do all the talking. I think possibly some of you have come from different backgrounds, but I mean, the way that I tend to think about this is what functional approaches can we do at scale so that we can sort of screen across lots of different, because you have tens of thousands of effects, right? So what you can evaluate, a high throughput, and then, picking a few examples, and then the way in which you'd functionally validate that is going to be very specific to the mechanism, right? I mean, without wanting to offend immunologists in the room, I think there's an interesting challenge of cell, what's the cell type and cell state that you clash up against a lot as well. So we have no. I could, if people start going to throw vegetables. So in the interests of I guess coming to some of the things we might be able to work on as a community. So there was some comments around how we might integrate disease or phenotypic, high level phenotypic information into our genetic studies. And I would say as a community, thinking about ways to standardize that between cohorts is going to be really critical. We're probably pretty good at doing that at a genetic level, like at the DNA, we've got well-established techniques for doing that at DNA. I think we're emerging to do that with single cell RNA sequencing, although there's still some way to go. But you've got a clinical phenotyping is a whole other kettle of fish, right? And having approaches to standardize that is definitely going to be important. And I think that definitely aligns with a lot of the things that HCA are keen to do. I mean, it was clearly identified by Sarah and Aviv on the morning session yesterday that's a focus moving forward for phase 2 or whatever the next phase of HCA is, where they need to focus on diseases. So we're going to have to think of ways to standardize that in a way that is represented as succinctly between subcohorts as possible. So Shyam, I don't know if we should add that as a feedback action item. By the way, he is making notes on his phone, not texting people, in case you're wondering why he's
a little both.
Both. All right, thanks.
Well, I've already added that. So thanks for bringing that up. The real power in integrative analysis across HCA Asia and across HCA comes from standardizing protocols. Very hard, easy to say, very hard to do on a global scale. But the point of meetings like this is to at least reach out to a few people to say, okay, I can't standardize with the whole world. Can I standardize with you, and you? And then we write a paper together. That's extremely powerful. And you can do things that are much bigger than what you can accomplish in your own lab. Certainly, IDA benefit is number of members of IDA over here, including Maureen and Bosyang and Fakrul, and hopefully Lan soon, people from Ponpan's lab and Rioya and so on. we benefited tremendously from the fact that we met each other at Human Cell Atlas Asia meetings. And then we got together and we just standardized our sample collection protocols, standardized our data generation down to, which lot of cells, control cells we will order from the vendor across countries, and then standardize the low-level data analysis and so on, made a huge difference in our ability to do transnational QTL analysis. So definitely I've made a note of that as one of our recommendations, but the recommendation makes sense only if you find each other, find your collaborative partner, and harmonize your methods with those people, right?
Actually, Shyam, do you think that aligns with the second comment at the top here? Because, one of the things that's, certainly I think we discussed, right, of interest is, what's, in the genetics community, we know a lot about how allelic effects, whether it's a GWAS or EQTLs, will vary in different cohorts based on genetic ancestral diversity of those cohorts, right? I mean, that's been a huge focus in the genetics community for a few years now. But with a single cell transcriptomic data and indeed epigenetics, you're going to have lots of sources of variation that are due to technical, environmental variation. And if we wanted to try to standardize with disease status, you kind of have those two things confounded, right? In most cases, you'll have a heavy confounding between disease subgroup, lab that is collected in country that the lab's in, the population group that it's been recruited from. And so if we're trying to think of ways that you can standardize the protocols for even naming disease subtypes or statuses, that would be required to address point 2 there, right?
Very much inclusion, exclusion criteria. Yeah. That's an excellent point. Even then, like you said, the clinical standards for diagnosing a disease can be different in different countries. But all you can do is harmonize to the extent possible. But there are dramatic gains to be had in statistical power if you harmonize your methodology, because there's some point at which if your data are technically really different, you will just spend forever scratching your head and worrying that this is a batch effect rather than a genuine biological difference. You just won't have the confidence to try to publish that result. It's definitely a qualitative difference that you get.
Drew, I'm gonna throw a question to. Sorry, I'll come to that in a minute. Those of you in the session yesterday would have seen Drew's been working on mapping genetic ancestral diversity for the bunch of the cell atlases that are publicly available at the moment. And so you've been thinking a lot about how this diversity might lead to phenotypic variation in cells. I mean, do you have a perspective on what this community can do using some of these standardization approaches or metadata that's collected on cohorts? Get a microphone now.
Thanks. Hi, I'm Drew. I'm a postdoc in Joseph Powell's group. I feel like those are slightly two different questions, right? We have ancestry and then we have all sorts of other kinds of metadata.
Yeah.
But I think you're right. I've been thinking a lot about all of this different type of metadata and how we can use it to understand variation across different samples across the world that have been collected. And I think that's really important. So one of the things that I will shout out here is if you are submitting data to HCA, or CZI, I know it can be a pain to fill in all of those different columns, but it does actually help people who are maybe looking at that data later on. And I think there are some good efforts at both HCA and CZI to help standardizing with these columns so that these data collection areas so that we can use them for these types of studies going forward. I'm not sure if that's exactly the direction that you were thinking for this question.
No, it's good. And an excellent call out as well. I think, there's efforts within, the portals that collect and share data and then the data ecosystems oversight committee. Is that the word of our committee?
Yeah.
Try to standardize some of this. Sorry.
I might take advantage of it having the microphone in front of me. I feel like there's kind of two broad areas of questions I've been hearing from the group. One is, how do we actually do EQTLs? And it occurs to me that we have maybe enough experience in this room and in this region that we might want to think about starting a monthly meeting where we can provide seminars or also just help for people maybe who are coming up against a wall and haven't had experience in this area. And it helps connect people who might be doing similar things and have the ability to look at different dynamics. And I think that this might be the right time. We've had enough people who have been doing work in this area and a lot of new people coming to the table. So I think it would be great. And then I think the other group of questions that I'm hearing are, now that we have a lot of experience in this area, how do we work together to combine our unique efforts together and be able to look at the data collectively, which is, I think, a lot of the questions that we've been trying to address. And I think that would be great.
Yeah, excellent. Thank you, Drew. Sorry. Yeah, you can take the mic now.
Just to quickly address Drew's point, thank you so much for bringing up metadata. None of this will be, all of this requires harmonized metadata. And within the Human Cell Atlas, is some people, Chloe, our keynote speaker yesterday, being prominent among them, who have worked really hard to come up with a metadata spec for the Human Cell Atlas. And so she's come up with a tier 1 metadata spec. She and many others have come up with a tier 1 metadata spec, including the HCA data wranglers, the CZI wranglers, and so on. That will be shared across all of HCA and has already been rolled out, as you know, to some of the early buyer networks. Within the diversity task force, we've come up with a human diversity metadata spec, which will again be rolled out in January or early February. And that will also, hopefully everyone working in this space will use that as a bare minimum of metadata harmonization. And that will be hugely empowering for us to be able to work together. And your other point was about
doing a monthly seminar to help
yes.
us all get together on a more regular basis.
That would be fantastic. I already know who's going to do the organizing.
The person who suggested is typically the person who raises their hand to do
volunteered,
it. That sounds nice. I was just going to comment on the part about standardization of metadata. So I think if you're coming in and starting a study where you're planning on what you're going to be collecting, it would be really helpful probably to go to HCA, see the types of metadata that would be helpful to include so that you can make sure that you're collecting as much would be helpful for both your study, but for the community as well as possible. And some of those pieces of metadata you won't be able to collect, of course, but I think if you at least take a look at that list at the front end, it would be also very helpful. So just a bit of a shout out for people who are generating data.
Yeah, again, I must confess that I actually have not done any EQTL analysis myself, although I've dabbled with or generated some data in the single cell data in IDA. But I've thought about this problem, and I think the problem needs to be teased out in a simple fashion because there are just too many variables. We're talking about EQTLs, and we have data on single cells, let's say RNA-seq data. We can first subset the cells and ask ourselves in which particular subset do we find an EQTL. That will require a power analysis, which is completely different from if you're trying to find a EQTL that will affect multiple cell types. So the power analysis will be extremely different when we consider how you're going to do the analysis. The second is that there are population genetic variations, and you need to take the if you're doing it in one particular population, your sample size will be of some kind to attain a specified level of statistical power. When you're bringing in variation in the genomes of these individuals through population genetics, that will require another. So I think the null hypothesis, and Shyam and Joseph, both of you are appealing to us to come together in some fashion. I think the null hypothesis needs to be very clearly delineated. Otherwise, we will always be underpowered. In any case, single cell studies are always underpowered. We'll be pathetically underpowered, and therefore the conclusions that we draw may not be tenable after 2 years when sample sizes. So I think we need to be a little bit careful, sort of lay out our questions clearly, and also understand the turf that we are talking about more clearly than what we are discussing right now, I think.
Yeah, I mean, I definitely agree. I think it's a little challenging sometimes because in most instances, whilst we've come together as a HCA community, many of us have generated cohorts for different funding mechanisms for different reasons. And so there's the logical point of, well, you know, is there a question that the community in this room can focus on? And I think it's probably outside the scope of this meeting to find out, but obviously, hopefully, it's goodwill to come together to think about ways which we can, address that as clearly as possible. You look like you're about to say something, but
Yeah, so as Joseph said, we may not agree in the next 12 minutes on what we're all going to collaboratively work on. You have a few more hours to make up your minds. But it's IDA started at one of these meetings, and then we just got together at a table, just like those white tables over there. Wong Yang, Jay Shin, and myself. And we said, okay, this is what IDA is going to be. And I'm hoping that conversations like that will happen during the breaks over here.
in the now probably 11 and a half minutes remaining, can I circle back to the question around spatial genetics? And what we're particularly referring to here is that, you know, many of us are generating spatial data on often much smaller cohort sizes, but still decent numbers. And is this an avenue that we can collaborate and come together to be able to evaluate how genetic effects are operating in solid tissues? And it's obviously of interest because there's a huge bias for single cell genetic studies in tissue systems that are easy to access, PBMCs and cell systems and so on and so forth. But yeah, again, so I don't know if anyone has any views on this. I'm kind of, maybe we can just get another. So like, who's working with generating spatial data at the moment? Again, hands up. And maybe we'll just flip through again. I mean, so what sort of sample sizes are you guys?
So, so my name is Murim Choi, and I'm in Seoul National University. We have been working on single cell EQTL of 50 cohorts of NAFLD, so half controls and half NAFLD or NASH. And we are putting the same set of samples on the Xenium panel.
Fantastic.
So we are hoping to get some data soon. And what we are wishing to do is trying to get not only the gene expression data, but the liver has a robust structure. How these robust structure are, you know, apparently spaced in the cases. So
yeah.
I'm very open to any kind of you know, algorithm person who can somehow quantify the spatial defects in the liver.
Yeah. And other people are working on spatial cohorts. What's
incredibly exciting, Maureen. Yeah. So just quick question. How big will your Xenium cohort be? 50. Brilliant. Very exciting. There were a few more hands up about people.
Hi, everyone. My name is Bailey. I'm from the University of Sydney. Our group is generating spatially single cell level spatial transcriptomics from histology data using deep learning methods. So that gives an opportunity to look at spatial genetics with a big cohort such as TCGA cohort. So once we generate spatial transcriptomics, then we have cell types, and we could look at the spatial patterns of each patient, and we could look at immune infiltration of each patient. And I think immune infiltration has been looked at using bulk RNA-seq. So I'm really wondering how spatial transcriptomics can capture immune infiltration compared to using bulk RNA-seq, how similar they are. And so immune traits always are driven by genetics. So we can run GWAS on spatial pattern, which can captured by some spatial statistics. So with TCGA data, they have 10,000 patients across different cancer types. So
that's super interesting. So is the approach there that you've got the histology slides and you're imputing
Yeah,
from
we
the
are
imaging data
imputing.
to transcriptomic variation
Yes.
with some degree of noise.
Yes.
But yeah, okay. Which might be overcome by the huge sample size.
Yeah. And another question is how to identify QTLs that genetic variants that affect spatially variable gene pattern.
Yeah,
Yeah.
that's one of the analytical questions
Yeah.
that we were thinking was particularly interesting. And I think the nice thing with the infiltrating cells is that, there's now large enough data sets, which many of us have generated on circulating immune cells. And so we have a good idea of what's circulating, the genetic effects, and we, are relatively good at classifying those cells. And if you have the relevant data spatially, you can work out, what cell subtypes have infiltrated and maybe what state shifts you've got if they've infiltrated the tissues. But, so that's interesting. Yeah, sorry.
Fantastic, very creative idea. Just a curious question. Does TCG have genotyping data available? Yeah. Okay, very good. And then coming to what you can infer, we're also doing some histology-based expression prediction, and Xenium data are very useful for training those models. What we suspect, this is not a mature field really, it's very early field. What we suspect is that from the histology, you can more effectively capture cell states than the precise expression of individual genes. So what you're getting is some embedding, some low dimensional embedding of the histology for that cell or that piece of tissue. And that embedding is certainly lower dimensional than the number of genes in your sample. And if it's lower dimensional than the number of genes in your sample, then maybe cell states are a better target. So my suspicion is that when we try to do individual gene EQTLs from histology inferred expression, we're actually just looking at some gene-gene correlations, really. We're looking at a meta-gene rather than individual genes. That's my hypothesis, but I think it remains to be tested. There were some more people doing spatial.
Yeah. Not actually QTL. I'm just trying to setting up different lines of a cohort, mapping the aging process. So basically the healthy control. So actually we are just generating data from the liver across ages and genders. So I'm trying to understand single cell and spatial omics. Yeah, so trying to understand how the QTL could be useful for understanding how the different vulnerabilities of agings, and as well as how these genetic variants are affecting, for example, the metabolic state of the different individuals. So that will in turns affecting the state of the individuals. So yeah, so basically that's the thing that I'm working on.
I'm conscious of the time. Is this the so any moment we're about to be swamped by
4 minutes.
people?
We have 4
we got
minutes.
4 minutes, have we?
Yes.
Oh, right. Okay.
Just to capture that, did you say it was a liver cohort? Healthy. Okay, so healthy liver aging. And what is the spatial technology? Is it StereoSeq? Okay, and how large, if you don't mind my asking, is it? 50. So, okay. So if people didn't hear, there was around a cohort of around 50 healthy liver aging using StereoSeq. That would be remarkable.
All right. So I think in a moment we're going to be swamped by people coming to find a spot to have their lunch. So we might wrap up. Shyam, seeing as you've been making notes and texting people, could you maybe just take a couple of minutes just to summarize, I guess, a couple of the key things that we hopefully have wandered towards as a group? We're going to summarize a little bit of this and share it after the lunch. But is there a couple of points you wanted to make just to wrap up?
Yeah. So we'll have, as Joseph said, we'll have a report back session later today. Please join that. All of the breakout sessions will report back. I think we have really achieved a lot in this breakout. So thank you so much for everyone who came and everyone who shared their future research directions. I hope you'll be rewarded for that with new collaborations and new ideas. We want to identify opportunities that people in Human Cell Atlas Asia can seize and make use of. So certainly studying, before we come to that, I would say, one gateway to all these opportunities is harmonizing protocols, harmonizing data generation protocols, harmonizing data analysis protocols, as Drew brought up, harmonizing metadata, which the Human Cell Atlas can be a nucleus for, right? Including the metadata. If we do that, then we can work together, and then lots of questions we can ask, which is, for starters, simply studying statistical power determinants for single cell and spatial QTL analysis. And Ryuya already has some exciting work, which we look forward to seeing. But I think it's an exciting field, and we certainly need more of that work. And then coming to actual, and then another enabling thing. So Drew mentioned hosting monthly or perhaps quarterly meetings, and she will email all of you soon. So, but it's a great idea because this should not be a one-off. This conversation should continue. And then what are the scientific questions? So some of them, Joseph. Okay. There you go. Joseph has already listed in his slides. So I think leapfrogging all the way to the most ambitious thing, right? Spatial genetics is a frontier. Let's jump on it. Let's leapfrog many of the other questions. I think if we don't do it now, if we don't right now, as Murim said, the methods don't exist. That's fine. That's the time to jump on a new field, right? And as you're working on it, you've developed some crude methods, and then someone will publish a paper here, you'll refine your methods, someone will publish a paper there, you'll refine your methods. But if you start early, you'll be in the best position to capitalize on that. So I think that's a huge opportunity for HCA Asia. You've heard about some of the people generating cohort scale spatial data. We are doing that as well for cancer in my group. I hope that we can come together on that. So definitely spatial genetics is another big opportunity. Then comparing QTLs across populations and across conditions meaning healthy versus disease. Another thing I hope people will come together in the breaks and work on what are the effects of geography and ancestry and environment on QTL effect sizes. And yeah, have I missed out anything?
I think that was the key stuff.
Okay. Thank you so much, everyone. And look forward to continuing the conversation. There's been a fantastic session.
Thank you very much.
And thanks to Joseph for preparing all the slides.